So the next speaker is Sabine Fischer. Yeah.
So, hello everyone. Nice to see you. I would first of all like to thank the organizers for this fantastic idea. So when I saw the title of this conference, AI and 4D cell physiology, I thought this is really something I would like to go to. And I'll tell you why in this talk. So first of all, to start off with, what do we do in my group? So I'm a mathematician by training. We don't do any lab work, just computers. But what we're focusing on is the link between morphology of a multicellular system and its functionality. So we're all interested in everything where space comes into play, spatial interactions when they are important. And for all our projects, we have collaborators. We have experimental collaborators, experts in the fields. And then what we do is we use this systems biology cycle that Aviv already showed at the first day. We do the quantitative data analysis to then move into our mechanistic models. And in this case, mechanistic in the sense of what a physicist would think of. So I'm not talking about molecules, molecular interactions in these cases, but we're actually focusing on cell-cell interactions or at the tissue level also, modeling of whole tissues. And one project where we've successfully applied this is mouse development. So we already had quite a nice introduction, so I don't have to say too much now. We're a bit earlier, so we're at the preimplantation stage. And the question is here, how do the cells decide whether they become embryonic tissue or extra-embryonic tissue? So we're at the inner cell mass here at the middle, and the cells at the beginning, they express both NANOC and GATA6, so these two transcription factors. Then they downregulate one or the other, and at some point you will have this nice separation where these cells later on become the embryo and the other ones become the yolk sac. In addition to these mouse images, what we also have is we have this in vitro system, ICM organoids. So those are mouse embryonic stem cells that are induced to, again, express GATA6. And then we can see similar patterns as we see in the ICM. Now, what people used to do was they mostly looked at the ratios between these two cell types. So how many NANOC positive cells, how many GATA6 positive cells, what's the ratio? But we were asking the question, what happens if we put space into all this? Do we get out new information? So how do we do this? The images I was showing you are just single slices, so they are all confocal images. So we have the 3D stack, so we can do the 3D nuclear segmentation. And then what we like to do a lot is to represent tissues as graphs. So here we have each node is a cell, and then each edge, in our case, means that the cells are neighbors, so they have direct membrane contact between them. And the nice thing about the graphs is that you can use all these graph analysis tools that you have. It's relatively straightforward then. Okay, from the graphs, we can also get more information because then we have not just the information on the individual nuclei, so their position and the expression levels and so on, but we can also quantify the neighborhood. So we then know which cell is next to which other neighbor, and is the other neighbor also NANOC positive or also GATA6 positive, so all these kind of information. And with these neighborhood features, we can then do spatial statistics. And here I'm just showing you one of the measurements that we're looking at. And the one relevant for today is the Morans index. So it's a spatial autocorrelation method. And basically what you need to remember is that it goes between -1 and almost 1. And if you're at -1, then you have more of a checkerboard pattern. If you are close to 1, then you have a separation. So it works well if you have two types of cells or objects in your system. So now with these kind of data, we move into our modeling approaches. And the top kind of data is something that we can put directly into the deep learning models because we have this graph thing, and that, again, gives us a native translation so that we can go directly to the AI or the machine learning models. The spatial statistics is something that we need if we want to go towards dynamical systems and these cell-based models. So there we have to do a bit of a detour. Okay. So we'll start with the first part because I said we wanted to put space into the game, and we were asking, is it actually relevant? So we decided we ask a machine learning model that, because if there really is a connection between the cell and what the neighbors do, then I could take the neighbors and asked whether I can predict the fate of the cell that I'm looking at. And this is what we did. So we trained, it's an MLP in this case, to try to predict the fate of this cell in the middle. And we first did all the analysis and all the sort of the ground truth that you have to do. We did the manual labeling, and that all worked well. So in the end, we went into our experimental data, and we did this for the ICM organoids, and we realized it does do it. It's not extremely great. We're at 70% accuracy, but still it's encouraging. So from this, we concluded that there must be something going on, and the spatial neighborhood must have some sort of relation. So that encouraged us to go into the cell-based modeling. And again, it's nothing. So the principle is something that people have used before in this context. So what we're assuming is we have this gene regulatory network. We have two transcription factors that inhibit each other, autoactivate, and then we have a signal from the neighboring cells, which inhibits one of them and activates the other one. And the idea how we implement this is that we have these promoter binding sites, and U and V have the same binding site. So this is how they inhibit each other. Only one of them can bind. And the signal helps V to bind. And for those of you who are interested in the equations, you have a production term for the transcription factors, and you have a degradation term. And we're basically counting how often can you have U binding versus all sorts of configurations that you can have. So what's your probability of U binding for this promoter site? And we're doing the same for V. And in there, one interesting parameter is this delta epsilon x, which comes up later again. This is the difference in binding energy between a bound state and an unbound state. Right. So the nice thing about these dynamical systems is they have these Waddington landscapes, basically, that everybody, not everybody, but a few people have been talking about during this meeting. And for our model system, because it's easy enough, so here we just have these two parameters, but in as many cells as we like, we can actually draw these landscapes. So what we know is that they depend on the signal. So if we have a low signal, then the cells will go to this U state. If we have a high signal, the cells will go to this V state, and then there's going to be a transition in the middle. And these models give you what people were asking for earlier, yesterday, for example, that you say, okay, if I have my cell somewhere here, I know exactly where it's going. So in these models, we have all this information, but now we, of course, have to link them to the experiments. So for that, first, I have to tell you a bit more about the signal. So we have to make a choice of what our signal might look like. And what we did, and this is new, so this is not what other people did in this context, we decided on a distance-based signal. So the idea is, in this case, that each cell interacts with any other cell in the system, and this interaction scales with distance. And we have the scaling parameter Q, and that is between 0 and 1. If it's close to 0, then the cells mostly talk to the cells that are nearby. If it's large, the cells talk to most of the other cells in the tissue. And then we can check what kind of patterns we can get with these. And as we've just heard, our brains are much better with 2D. So the simulations can be equally done in 2D and 3D. And for comparing with the experimental data later on, they will be in 3D. But for you and me to capture this easier, I put the 2D results here. So what we see, first of all, for our scaling parameter is that if the cells only talk to cells nearby, then we get something like a period 2 pattern, checkerboard pattern. If we increase the range that cells talk to, then we get more of a local clustering. And at the end, we can even get an engulfing pattern. And here's our binding coefficient, this energy coefficient again, the difference in binding bound and unbound. That helps us so that we can actually also tune the ratio of the different cell fates. So this is nice. So we have our model. We see that it can generate patterns similar to what we might have seen in the experiments. So we started comparing them to what we have from the experimental data. As I said, people haven't done these distance-based approaches yet. The usual thing is nearest neighbor. So you just integrate what the nearest neighbor does. It's usually an average. And then, of course, for the mouse embryo, we also needed to include cell division. And then we looked at the measurements. So we, again, calculated this Moran's index for our simulations. And the first thing we see is that this nearest neighbor one gets the smallest values. And also if we put in cell division, in any case, then it will always be a bit higher. So that is reasonable. Cell division gives you clustering. We've also shown that in other studies. Now, interestingly, if we compare these results to the embryo data, then these are the ones that fit. So what we can take away from this is that the nearest neighbor signaling isn't enough. So the cells in the mouse embryo don't just talk to their direct neighbors. They need to talk to cells that are a bit further away as well. How far, we're not quite sure yet, but the nearest neighbor signaling certainly is out. So as I said, for this one, we had to take the detour via the Morans index, but I also said that we could use the graphs to put them directly into the artificial networks. So here, what we did, and again, this is in 3D, I'm just showing it to you in the 2D. We used graph convolutional layers, so a graph neural network, and that can just take this whole graph that represents our tissue, and then can process it. And the task we gave this one, in particular, was to predict us this dispersion parameter. So that was the parameter that tells us how far the cells talk, how far that goes. And again, we did the manual label. So we need to train this with our simulation data because, of course, for the experimental data, we don't have a ground truth for this Q. So what we can do is we can train it with the simulation data. For the 2D case, we also, again, did manual ground truth and compared the model. It was much better than we as humans. In the end, we then applied it to our experimental data, again, of the ICM organoids. And what was quite interesting to us is that both for the 24 hours, but also for the 48 hours, this dispersion parameter was rather low. Now, before that, for the 48 hours, I would always have thought that they are fully sorted. And if we would have fully sorted, then you would expect a Q that is much larger. So that was a bit puzzling. And I remember actually my student saying it's not working. Because he said, look, I have these values and they go only up to 0.5 and I was expecting 0.9. It's not working. So what we did to also look at it is go back to the cell-based model and also look at the cell-based model. And here we have the 24 hours and the 48 hours. And again, so these different colors give you the different Qs from 0 to 0.1 to 0.9. And the black ones are the experimental data in there. And again, we're only sort of up to 0.5 even for the 48 hours. So this also confirms that there is some interaction with the neighbors further away. Now to summarize, what I wanted to show you today is that we can use these graphs very nicely to represent the spatial relationships. I also wanted to show you that to look at the 4D, so space plus time, I think it's quite useful to also include mechanistic, in my sense, mechanistic or dynamical systems, because there we have the time in there. So we can either generate the data for the training, or we can maybe use it to restrain our models, our AI models. I've shown you that with this long-range signaling, you can get this engulfing pattern. People have usually used, cell sorting could also give you that pattern. This is an alternative to it. And finally, I've shown you that for the mouse embryo, it seems that nearest neighbor signaling isn't enough, but you need to go further. Right, so this is the people who did the work, and I'm really grateful for the collaboration partners, the funding bodies. And if you want to get to know any of those people, you're a PhD student or a postdoc, and want to talk more about our work and see our beautiful city in Wuerzburg, then apply for our visitors program. Thank you very much for your attention.
Yeah, so great talk. So I think I have two detailed questions. So the first question, so you mentioned that there's a difference from the nearest neighbor graph and the distance-based graph, but actually they are kind of not that different, right? So yeah, so just like a K, so if we can select a K, that is just in the, some kind of the radius, so the K is just there. And the second question is that you are using the graph convolution network. That is probably the first generation of the graph neural network. So have you tried to use some maybe the attention-based things? So probably this attention can kind of give you another different layers of the information to make your results interpretable. Yeah, thank you.
Thank you for your question. So for the first one, to clarify, the graph is not different. So the nearest neighbor is what we include in our dynamical systems. The graph will always have edges between the nearest neighbors, so there's no difference there. It's just what do we include in our differential equations where the signal comes from. The second question, no, we're currently working on continuing on this graph analysis methods, and we haven't used any further. We're not there yet, but we're on it. And thank you for the suggestion.
Thank you.
All right, one last question.
All right, so I love it. I want to extend the discussion on your comparison between the signaling-based and cell sorting-based mechanism for this engulfing pattern. Can you tell me, like, if you, let's say, consider the cells can now move around a little bit, can you now get kind of like better sorting pattern or more segregation with kind of like a very close distance just by talking with the nearest neighbor, for instance?
So this is something that we're actually looking at the moment. So we've started changing these kernels basically that describe of how far the cells can talk to each other, and also the weighting of the systems. And we've now included the cell sorting within there. And depending on how you do the weights, so which would represent how does a cell calculate between its neighbors, you might not get the engulfing pattern straight away, but you can then get it through the cell sorting. So yes, that also works.
All right. Thank you so much, Sabine. And let's thank all the speakers once more.